Hello everyone and welcome to another live presentation in Beckman Coulter's Life Sciences webinar series. My name is Lutz Erhard, currently working at Beckman Coulter as a marketing manager for centrifugation, and with great pleasure I will be the moderator for the webinar by Dr. Alexander Bepperling. Our speaker today is currently holding a position at Hexal Sandoz. There he is heading the AUC facility of Sandoz Novartis. Besides AUC, he is using various spectroscopic techniques like CDFT-IR, fluorescence spectroscopy, and is also using surface plasmon resonance. Dr. Bepperling carries his PhD in biotechnology. Previously to his work at Sandoz, he was working in science at the Martin Luther University Halle in the Department of Industrial Biotechnology with Professor Rudolph, and at the Technical University of Munich, which is one of the Germany's top 3 universities and part of the German Excellence Cluster. There he worked in the Biotechnology Institute led by Professor Buchner and was part of the Center of Integrated Protein Science Munich. Before we begin, a few housekeeping items. The presentation of Dr. Bepperling will run for approximately 35 minutes. After that, we will have time for a moderated question and answer session. Questions for this session can be addressed all times during the presentation. You can enter them after clicking on the Q&A widget on your screen. This widget should be green. Also, I want to note that the webinar and the Q&A session will be recorded and made available after the webinar, so you can focus on the presentation now and not on making notes. A final note, I ask you to take the time for a short survey about your background and experience with AUC. This will help us funding the right content level for future webinars and get funding for application content work. So I show the poll right now and ask you to answer those questions, easy questions, within approximately 20 seconds. So questions are whether you're using AUC and you're interested in latest updates as an experienced user, or whether you are using AUC as a beginner, or you don't have experience but you're using complementary techniques like SEC, HPLC, light scattering, and then whether you're working in a biopharma environment or in an academic scientific institute. Some more seconds here to close the poll. So poll results. So we have 20% experienced users in the call. A lot of the people here using complementary techniques. And we have a good split here, biopharma and academia, a little bit the balance on the biopharma side. So thank you for answering the poll questions. So now I would like to pass the ball to Dr. Bepperling, and I look forward for his presentation now.
Okay, thank you, Lutz, very much for the kind introduction and for giving me an idea about the audience today with the poll. Today I will mainly focus on the data analysis tools to achieve high precision when analyzing biopharmaceutical products, because we are mainly interested in minor species of about 1 to 2%, usually comprising of the total protein content. And I will not go too much into the practical details because they are outlined in a series of excellent publications from the people at Amgen, for example, Kelly, Arthur, Stoner, Gabrielson, and these guys. So I will more focus on the data analysis done in Sedfit, and I will also not compare it to the data analysis method of Borries Demeler's UltraScan. Okay, just a brief introduction. In AUC, you are basically spinning a sample and watching it. And it is all based on the pioneering work of Theodor Svedberg, who made a rather significant discovery about migrating objects in gravity fields about 90 years ago. And what he found out is that the velocity of a particle divided by the acceleration it experiences is constant. And the reason for that is that upon spinning the rotor and the particle picking up some speed, it gets into the balance of three forces: the centrifugal force, the buoyance force, and the frictional force. And all those forces are linked together and form a constant velocity, which is called the sedimentation coefficient, abbreviated as S, and it is related by the famous Svedberg equation to the diffusion coefficient and the mass of the particle. And what is useful for us nowadays is that every particle in such an experiment forms its distinct boundary. You may see it by eye, like here in this example with a BSA sample. You see at least two boundaries. Every component has its own boundary, as I mentioned. And in the trailing edge, there you find the degradation products, and in the leading edge, the high molecular weights aggregates, which we are interested in. You may not see them by eye, but they are deconvoluted by modern data analysis methods, which is greatly facilitated by using Sedfit. I will go to this in a few minutes. Just a brief look at the hardware for those who have no experience with AUC. The samples are filled in a pre-assembled cell, which is composed of about 20 parts, and the main part is here in the middle, the so-called centerpiece, which holds the sample and can be seen some like the column in an HPLC experiment. The sample is filled in there, sealed against the vacuum, and put into a rotor. There's a 4-hole rotor and an 8-hole rotor available, orientated perpendicular to the axis of rotation, and put into the rotor chamber. You see here the three currently available commercial optical systems. The fluorescence system at the left corner, available from Aviv in cooperation with Beckman, the interference optics, and also the absorbance optics, UV absorbance optics, which is mainly used in experiments because when we look at the next slide, this is the machine. The absorbance system is sensitive to chromophores, of course, mainly the aromatic amino acids in the analysis of proteins, or mainly antibodies, which is the main topic in biopharmaceutics, whereas the interference optics is not sensitive to chromophores, but records every contribution of the signal, including buffer components, and usually we don't have the possibility to use dialyzed samples, although interference optics has some distinct advantages, especially the high concentration range. I will show an example later on. Fluorescence optics is very useful for low concentrations and also for analysis of tight binding interactions, even in the subnanomolar range. But of course you have to label your protein, because currently the system is a solid-state laser with an excitation wavelength of 488 nanometers. And so you have to label your protein with a fluorescein-like dye, with the Atto or Alexa 488 series, for example, or attach EGFP, EYFP moiety to the protein. Okay, let's consider you have gathered your data correctly using the protocols outlined in the paper I mentioned in the beginning. So you equilibrated your rotor thoroughly in terms of temperature. You aligned the cells very carefully and gathered data at optimal wavelength. Then you may start a new instance of Sedfit and load your data in there. And what you see in the first beginning is in the upper window your raw data. and in the lower window the depiction of the fit data. Of course, now you don't have done any fits so far, and now let's initialize the parameter. When you choose the model, usually you choose the continuous of S distribution because it's the model-free description of the data, and set the parameters. I show you here an example for the previously shown data set. So it's a monoclonal antibody diluted to about 0.5 mg per mil, analyzed in PBS. So at a AUC ideal buffer with salt in there and at a moderate concentration. I prefer to set the resolution to 100, so the size distribution is cut into 100 slices, and the Lamb equation is solved for 100 species in there. This is usually sufficient if you look at one order of S, so let's say from 1 to 10 or from 2 to 20S, and this is also the maximum in my experience which you can get out of at a single speed. If you want to have focus on the more smaller aggregates or degradation products, you may lower the speed and analyze a different set of S values, but I prefer for the usual 150 kilodalton monomer antibody and its dimer, trimer, tetramer up to, let's say, 2 megadalton to set the range from 2S to 20S. If you have smaller proteins, you may set it from 0.5 to 10. For example, if you have cytokines or proteins like that in the range of 10, 20, 30 kilodaltons, we have some of them on the market, so there's also need for that. The 1.5 frictional ratio is a good starting guess for a monoclonal antibody. You want to check this and let it fit. You also want to fit the baseline and the time and radial invariant noise, so you always check on both. Noise contributions and also the baseline. You also want to fit the meniscus and let it float because it's correlated to the frictional ratio. I do not like it, although it's publicated, to fix the meniscus graphically and let only float the frictional ratio because both of them correlate highly together. So when you fix the meniscus, you force the frictional ratio to maybe an unrealistic value. Therefore, you don't want to do that and float both. The bottom can be fixed because the cell has a fixed geometry and you don't want to fit for the bottom in a velocity experiment, other than in an equilibrium experiment. I also like to set the confidence level for the F statistic ratio at 0.95 to standard deviations and not to 0.68 because this suppresses artificial peaks. And this is what you want to know. You want to know that the peaks you are finding after convergence of the fit, that these peaks are actually real and not fitting artifacts. Okay. The partial specific volume and also the refractive index increment and the extinction coefficient can be computed in Sedfit. You find it under options, loading options, in calculator, VBAR, the refractive index increment and extinction coefficient from protein sequence. It's pretty close to 0.73 for most monoclonal antibodies, so I'll leave it at this default value. And I calculated the buffer density and viscosity for PBS using Sedinterp, or if you have a complex buffer, you may want to measure it using the Anton Paar DMA 5000, or for the buffer viscosity, this Lavis 2000 ME, which is the predecessor of the Anton Paar BPN densitometer. Okay, if you want to have a defined S-space, but this is usually not the case, you can upload this, and also for big and broad particles, you may load up a log space grid, not a linear one, but for antibody aggregation analysis, this is usually fine. Okay, after you initialize the parameters, you may choose the meniscus and the fitting limits, and afterwards press run and check for spikes and also for coarse misfit of the parameters. The meniscus can be easily located right at the apex of the optical artifact. This is quite trivial, and the bottom you may leave it as it is. A little bit more critical is the fitting limits. In my experiment, in my experience, it's the best usually to look where the second scan enters plateau, and there set the first fitting limit close to the meniscus for aggregation analysis, because it enhances reproducibility. Here in between, so in this space here, the location of the fitting limit slightly influences the aggregate content you measure afterwards. If you go further into the solution column, it's quite constant. So I prefer for reproducibility to set it here at this position. If you have an interacting system, of course, you want to have it as close to the meniscus as possible to include the full data range. The bottom fitting limit is usually defined by the back diffusion. If you have a small molecule and a low speed, the back diffusion may reach quite far into the solution column. You can calculate this using the simulation function of Sedfit, but for a monoclonal antibody, which is quite large, at 50,000 RPM, everything is pelleted at the bottom and the back diffusion region is quite small. So I set it here to 7.15 centimeters and also leave it there. For the rest of the analysis, when I, for example, analyze multiple cells of one rotor, I choose the lower fitting limit here and leave it constant for all cells. And in the front, the upper fitting limit, I choose it, as I said, to the position where the second scan enters the plateau. This gives, in my experience, the most stable analysis of the monomer, dimer, and so on fractions. Then you may fit this and document it by integrate it and report it. I check always the RMSD, which should be in the range of 0.005, and you may track it over time. Although the absolute value is not that interesting, it gives you over time hints if your lamp gets dirty over time, or if you have a problem, for example, with your buffer, which you use for dilution. So I track this and also report it every time when I report an analysis. For the OD, there's plenty of literature available discussing the optimal OD. It's in the theory quite simple. The Eulerian absorbance should be 2, so I over I0, and the logarithm of it should be 2. So this means the logarithm, decadic logarithm of 2, which is 0.8686 and so on. So I use the 0.87 as a target at 12 millimeter path length, and this gives you an optimal signal to noise ratio and also the maximum signal, because you want to see fractions of the total signal. So you should go as high as possible before the optics is not linear anymore and the noise starts getting higher. So this is, in my experience, the optimal OD. And to avoid fitting artifacts, because when you're an experienced Sedfit user, you may notice at 0 or at the lower fitting limit and also at the upper fitting limit, you may have these peaks which shift always when you increase or decrease the fitting range. So for example, you have this artificial open peak, half peak at the end of 20S. When you increase the fitting range to 30S, it's still there. You increase to 50S and it's still there. This is just fitting artifacts. At the lower S range, it's baseline correlation and maybe also some non-sedimenting buffer background, and at the higher S range, it's simply spikes, because a spike is a steep single signal, and if it occurs twice in a run, which is quite common, you have created an artificial steep and high boundary with a high S value, and also if you fit it separately in UltraScan, for example, with a high F over F0. Therefore, I set a range where the protein is located, here in this case from 5 to 19S to 100%, and calculate the peaks respectively. So I click on the boxes and report the percentage species of, and calculate percent of total protein. You also should have a look at the S values to see if those values make sense, and also to check, especially for smaller proteins, it's not that trivial if it's dimer, trimer, and so on, because the fitted mass is of course dominated by the frictional ratio of the monomer. Okay, and when you do this, you may be able to quantify your aggregates quite precisely in a matrix-free environment, which is of course very useful, and also the reason why AUC is still used in, or more and more used in biopharmaceutical industry, because the precision may be lower compared to size exclusion chromatography, quite lower to size exclusion chromatography, but overall you have the advantage that you are matrix-free, it's non-destructive, and you can analyze it directly in formulation buffer. Here shown an example for an Fc fusion protein, and what you just may take home as a message from the table, you see that you can expect to quantify 0.1 and higher % of oligomers. Usually the LOD, LOQ is still under discussion, but you can say you can expect to be peaks above of 0.3, 0.4% to be really true. And the precision you may expect is also in that range. So 0.3 to 0.5%, in my experience, is the precision you can obtain in a triplicate measurement. Okay. What also came up during last month, but it's now fixed, is that when we did cross or multi-site calibration studies, or if you have more than one instrument in your lab, that the S values you got from different machines were quite different, and this led to a series of experiments designed and also published by the group of Peter Schuck to improve the radial and thermal and the precision of the temperature control of the machine by correcting it and calibrating it using external standards. So it started with a little problem, which is now fixed by Beckman, and ended up with a quite useful series of methods to improve the reproducibility and also the accuracy of your S values reported from different machines, allowing you direct comparison from lab to lab, or improving it, let's say, in this way. When we did our first experiments on the newly acquired Proteome Lab software on our machine, we saw that the S value shifted slightly of about 10%. Masses were always higher, about 170 kilodalton for a monoclonal antibody. And I talked to Peter Schuck, and he noticed that problem long time before, much earlier than me, of course, because he has quite big park of AUC machines. What he found out is that there was a timestamp error for the particular combination of hardware and firmware and software of the AUC, and he implemented a correction function located at loading options nowadays, and this checks the elapsed time from the timestamp of the Windows timestamp when the file is created with the omega square t, which is recorded in the header of the file. And my machine now has a new GUI and a new version of hardware. What you see is the correction factor nowadays is really, really small, only 1 promille of the time, so you can neglect it. But you may check your GUI and your software if you are really on the current state. If this is the case, you usually can neglect it. Otherwise, you can simply correct it using the Sedfit function. And what they also derived, and what I did on my machine, I show you an example here, is to check the rotor temperature and compare it with an external thermometer, a calibrated thermometer, because the density and much more the viscosity of water around 20 degrees Celsius is very sensitive to temperature, because apart from 20 degrees Celsius, a change of 1 degree Celsius changes the viscosity of about 4%, so it's quite a lot. And there are these quite useful and also cheap iButtons, about 100 euros for all, for the data logging device and the iButton itself. It's NIST calibrated, and this model can store up to 4,000 data points with a precision of 0.06 degrees Celsius. And what you can do simply is put it into the resting rotor, close the rotor chamber, set it to 90 degrees here in my case, because I pre-equilibrated it for inserting the cells afterwards, or you may set it to 20 degrees, it's up to you, and record it over some hours. And what you see is, of course, the rotor cools down and approaches the temperature set at the console, here in this case 90 degrees Celsius, and ours has a temperature of 17.8, so it's a little bit lower. This is not a big problem, because you can simply plug it into Sedfit, Sedinterp, Sedfit. Most softwares can simply handle this and convert your S values to SW20, so water 20 degrees Celsius. What is also quite useful to do, and I'm currently working to get such a radial calibration mask at a machine shop in Germany, is to use a black and white alternating pattern embedded into such a mask, which is inserted like a 3mm centerpiece into the AUC cell. So you use 3mm ring spacers and the calibration mask just like a flat centerpiece, and what you record is the alternating black and white pattern. You know it's 1 millimeter black, 1 millimeter white, and you compare the pattern to the measured one and can derive a radial magnification correction factor. Usually this is also quite small, and I'm working to make such plates, and upon request I can send either you the dimensions, a sketch of this mask, or in a few weeks, I think, also upon request, a mask itself. Okay. If we look, this is an excerpt from a publication, at various machines Peter Schuck tested, you see there's quite a big range in the temperature, which can be easily corrected by applying the SW20 correction. And you also see there's an error, but it's not that large in the radial magnification, and the time factor is currently not a problem anymore. What is quite interesting to see, this is a BSA standard, living forever, so to say. Peter Schuck filled it into a cell and never disassembled it, and over 10 labs participated. And what you can see here in the beginning, the S range of the monomer was quite dispersed, and when you correct the S values collected from various machines and various labs and apply all those corrections, you end up in a very, very narrow range of S values, and also at a good fit for the molecular mass. Although this does not matter that much for aggregation analysis, because the overall size distribution is not affected in such a high, is not affected by those factors, because they scale the S values but not the distribution itself. It's really useful to enable those corrections, because it underlines the value of AUC as still being the gold standard for measuring particle or protein sizes in solution, without the need of introducing calibration proteins, so without calibrating S values by let-run molecular weight standard like in size exclusion chromatography. So I would recommend it to use it. And also to report, of course, S values and state whether you have corrected them or not. Okay. Besides the analysis of diluted protein concentrations at ideal buffer systems and with a nice experimental conditions, you also may want to have a look at protein formulations in final dosage forms, which have nasty excipients in there and a high concentration and represent a real non-ideal AUC system. But still, you can obtain, and this is an example of a stressed monoclonal antibody at 8.0 mg per ml, which is the final concentration in the infusion bag. You see that you can quantify relatively precisely the P50 and P100 fragment, which is often hard to quantify in size exclusion chromatography because it forms a shoulder only to the monomer. You also can quantify dimers and also higher oligomers, although you may lose some precision at the higher oligomers because the baseline is ill-defined when you have a gradient formed by excipients. But it's still useful to see what is really going on in your formulation, also to see whether there are gross changes of the monomer-dimer content upon dilution or buffer exchange. Okay, to sum this up, AUC covers a large size range. I told you in the beginning that depending upon the size of your monomer, you should adjust the fitting limits. You may also adjust the speed. When transforming these 3,000 to 60,000 RPM to kilodalton or megadalton, to give you an idea, this corresponds to 1 kilodalton to 1 megadalton, 100 megadalton. But as I said in the beginning, you are limited with one speed, and currently you can perform AUC experiments, velocity experiments only at one given speed. This limits you to 3 orders of magnitude within one experiment. So if you want to have a look at, let's say, your monomer, dimer, and some higher oligomers, you may fill the cell as full as possible and run it as fast as possible. But afterwards, you may analyze it again, looking at higher oligomers and running at 20,000 RPM, for example, to look at the high megadalton oligomers or even submicron particles. You can analyze your samples in nearly any formulation buffer. So far, I encountered no system, and we have really nasty formulations from a spectroscopic perspective sometimes, which cannot be analyzed by either interference or absorbance optics. You may not have to dilute your sample. Of course, you should if you want to precisely measure aggregate content, but you may have a look at the undiluted formulation. And of course, the main advantage is you do not have matrix interaction. Okay. To underline this large size range, I show you here an example of 3 vaccine batches from our vaccines and diagnostics division. This were 3 different batches of one vaccine, and what you see here is that they had a considerably different high molecular weight aggregate content, although it was a really broad distribution. The overall active species were these high molecular weight species, and they all had a different potency. So this is really useful to analyze such size distributions in the megadalton range, which is really hard by chromatographic techniques. Okay. AUC is also able to cover a large concentration range. Here an example shown 100 picomolar EGFP and 1 nanomolar of an anti-EGFP antibody binding to it. And what you see here, you can clearly discern the boundary of the EGFP alone and the boundary in the binding experiment of the remaining free EGFP and the antibody forming the complex. So you are able to obtain KD values, even in the sub-nanomolar range. Peter Schuck recently published a paper where they analyzed an interaction with a KD of 20 picomolar, so really tight interactions. And on the other end of the range, you may want to have a look, and this is a protein which at 100 micromolar was dimer, at 300 micromolar was still a dimer, but upon shifting to 1 millimolar, it formed a 20-former. So at high concentrations, your formulation may hide some surprise regarding the oligomeric state of your protein, and this is accessible by AUC. And you also may be able to derive binding constants for really weak interactions in the millimolar range and associated to it the second virial coefficient. I'll show you an example afterwards. Okay. In the biopharmaceutical field, we usually deal with high affinity interactions. So interactions far too low to be analyzed in terms of KD by the absorbance or interference optics. But what you can do is, because you are far above the KD, and also if you have a high interaction, it usually has a very slow K-off rate, usually in the rate of 10 to the minus 5 or even slower. You may regard all of your peaks as constant, so not as a peak of the Gilbert-Jenkins theory, which deals with the interacting system and the prediction of interaction boundaries and reaction boundaries. So you can derive stoichiometries because you can regard the peaks as constant. And what you can also do is you give after you check your target, for example, here in this case TNF-alpha is binding competent, you may add an excess of your target protein and check whether your protein formulation contains any non-binding incompetent fraction. Here an example shown for 3 anti-TNF-alpha binding proteins. There you see you have one antibody forming very small complexes and 2 other ones forming quite high molecular weight complexes. And when you have a closer look, this is the antibody 1. You see free TNF-alpha, free protein. You see complex 1-to-1 forming, and also at higher concentrations a 1-to-2 complex forming. This is a good basis, for example, if you afterwards analyze your proteins via ITC or for tighter interactions using the plasmon surface resonance to impose a model. Okay, you also may be able to elucidate complex binding mechanisms, although a KD, of course, is difficult to assign for those reactions. You see here for the antibody 2 in the previous slide, this should be free TNF-alpha, this should be free TNF-alpha, and this should be the free antibody, sorry. And you see 1-to-1 complex forming, 2-to-1, 2-to-2, and so on. So you can really assign different complexes, and also you may be able to record subtle batch changes, because this pattern, of course, is really a fingerprint-like pattern, and it's sensitive to buffer changes, for example. Okay, now let's switch to sedimentation equilibrium for the rest of the time. In those kind of experiments, you do not pellet your protein at the bottom of the cell in a sector-shaped double centerpiece, like I showed you in the very beginning, but you use 6-channel centerpieces with 3 sample compartments and 3 buffer compartments, and centrifuge it at a series of lower speeds until the protein comes into, or the solution comes into equilibrium between the sedimenting force acting towards the bottom and the buoyant force or the diffusion force acting towards the meniscus of the cell. And the distribution follows a single Boltzmann distribution, which can be fitted to a single exponent, where the molecular mass or the buoyant mass is the only unknown in this equation. And if the masses are known, then you may link those masses in a case of an interacting system by a association constant. And what is really useful is that such a sedimentation equilibrium experiment is easily set up and gives you data which correspond to a full titration series, for example, in an ITC experiment, although I love ITC. Here, for example, if you have an A plus B goes to AB system, here at the meniscus, you have the lower concentration, so you have more free A, more free B, and less complex, while at the higher concentrations, at the bottom of the cell, of course, you have less free A, less free B, and more of the AB complex. So each point of those concentration series correspond to a single titration point. And this gives you a very robust KDs, and you can use this to screen, for example, binding, especially low binders with a micromolar affinity, which may not act as pharmaceuticals alone, but to check, for example, if domains are expressed correctly or if they can assemble. I show you here an example. I used this to screen the affinity of an isolated heavy and light chain variable domain of an antibody, where we mutated different sites at the binding interface. And what you see here is we mutated the light chain and held the heavy chain constant. And what you see here is most of the mutants were not that dissimilar to the wild type, but when we mutated a proline residue, the KD got much worse, from the 3-digit nanomolar range to about 15 micromolar. And you also can see it by eye. This is the high affinity interaction with more complex forming, and this gradient is more shallow with more free A, free B in there, so barely interacting. What you can do apart this binding screening is you may also want to have a look if non-protein compounds bind to your protein. For example, you look for detergent binding in formulation, because this is also sometimes a question which comes up. Does the detergent in your formulation interact with the protein itself? And what you can do there is you increase the density of your solution by adding D2O or A2O18 to make just the solution more dense, and to look at the buoyant molecular mass versus the solution density, and the intercept at 0 gives you the partial specific volume. For tween alone, tween AD alone, this was only proof of principle. This is quite trivial. You also could have looked it up. But in another example, we looked at EPO in formulation buffer and did the experiment again. And when we extrapolate to 0, we saw a V-bar of 0.6965, which is the V-bar of EPO alone, meaning that no detergent is binding to the protein. Here just an example to show you when protein lipid interaction occurs, then you are also able to fit the number of detergent molecules bound to your protein. Here 3 different detergents, Choline detergent, SDS, and an octylglycoside, and binding to apolipoprotein A, an N-terminal truncation mutant. What you see here is the number of detergent molecules bound to the protein vary with the kind of detergent you use. So this is sometimes, if it's an interaction with between protein and lipid, quite useful, because then you can give an estimate how much of your lipid or detergent is bound actually to your protein. Okay, I also promised to show an example of the second virial coefficient, which is thought as to be a predictor for protein aggregation, not the only one, but an important one, which is a measure of the non-ideality of your antibody solution, indicating if the interaction at high concentration is repulsive or attractive. And here I show you an example of two antibodies, one with a very repulsive interaction and another one with a nearly neutral second virial coefficient, so a really low positive second virial coefficient. And what you just do is you analyze in a velocity experiment at one single speed, not at a series of speeds, the apparent molecular mass and plot it against the protein concentration. And the slope is a second virial coefficient, so quite simple and not easily accessible by other methods. You can do light scattering, you can do self-interaction chromatography, but AUC is really straightforward and quite fast done. Okay, and this also correlates in this case to the aggregation behavior. We have here the antibody designated as AB4, which has the high repulsive interaction, stored at 40 degrees Celsius for 6 months, and maybe a little bit hard to see, but you have barely forming aggregates. So it's still exclusively monomer, while the AB5 forms under such conditions up to 25% of dimer. Okay, now coming to the end, I will show you a selection of special centerpieces which are also available at Beckman Coulter. They base on the observation that at above 5000 G, gravity breaks the surface tension of water in small capillaries, and then you can transfer liquid between two sectors in your centerpiece. For example, if you consider a synthetic boundary, this is quite useful for the determination of the extinction coefficient and vice versa of the concentration. I will show you an example of determining the extinction coefficient in the next slide, which is a quite tricky task sometimes. In the beginning, you have your buffer in one channel, the protein here depicted as little red dots. Your protein solution in the other channel, you spin it at 10,000 RPM roughly, and then liquid transfer occurs. And what you immediately get out of this is a fringe pattern, which can be directly converted into mix per mil of protein, because for large proteins, the refractive index increment is fairly constant, because all those amino acid contributions cancel out, and you can convert it directly to protein concentration in mg per mil without diluting your samples. So you may analyze, let's say, a formulation at 50 mg per mil and get directly the concentration. If you use lower concentrations, and which permits also the recording of an absorbance signal, you can determine the extinction coefficient. I'll show you an example in a few slides. Also useful is a meniscus matching centerpiece, which layers some buffer through this channel to the bottom of the solution of your protein, and the result is an exactly matched solution column, which is very useful if you have an interference experiment with different interference experiment with a quite difficult buffer. Okay, there's also a band-forming centerpiece available. If you have a precious sample, you may layer it on top of your protein, your solution column, and have a look on the migrating band which migrates through the cell. Or you may want to have a look in an equilibrium experiment where your molecules are located in a density gradient, giving you an idea. For example, if you have free DNA, free RNA in your sample in there, for example. Okay, this is a brief example for determination of the extinction coefficient for a new project newly started in our lab. You see here the buffer signal in the interference as well as the absorbance optics. Red is absorbance, black is interference signal. Here's the baseline. And you also see the plateau of the absorbance and the plateau of the interference signal. You just subtract them and divide it by each other. So you have your protein concentration in mg per mil and the absorbance, which corresponds to this protein concentration, giving you the extinction coefficient. The good thing is you don't need the exact determination of the protein concentration. Okay, I hope I could show you AUC is a really useful and versatile tool for studying proteins in solution for the biopharmaceutical industry. It's non-destructive, sometimes very useful if you want to recover it afterwards. It's sensitive, it's an equilibrium method, nothing enters the cell, nothing leaves it. You can have size and shape and also mass distributions in nearly any buffer system. You may be able to determine S and D independently, and thus you can derive the mass. You can also enhance this, as I showed you, by using external standards to enhance the precision of S and D, and thus also M. Although the size distribution is not affected, so for quantitation it's not that important, but there are reasons to of course improve the accuracy of your measured S values. You can discern reversible and irreversible interactions, and you can predict aggregation by using the second Virial coefficient. You have a large concentration range accessible up to 80 mg per mil using 3 mm centerpieces in the inference optics. You may want to have a look at ternary interactions, which is difficult, for example, by SPR. You can simultaneously determine size, the concentrations, stoichiometry, KD, and K off in one sample. And you have no dilution, as I said, no matrix interactions, and a really sound theory for over 80 years now. Okay, this is the literature for the calibration procedures if you want to perform this in your lab. Thank you very much for your attention.
Thank you, Alexander, for your presentation. I found it very interesting. During the presentation, I have received a number of questions. We are short in time with the questions, so I would like to proceed as soon as possible. Mm-hmm. I grouped the questions a little bit regarding the topics. So I have questions about what kind of samples you can analyze, a little bit about concentration, and then questions about where in the process you place AUC. I would like to start with some questions about the samples that can be analyzed. Okay. Reading the questions, I will use your first names for data safety here. So I have a question here from Yang, who asked a question here that he's reading a paper about AUC, and this says that a protein sample you are looking at with AUC needs to be at least 95% pure by SDS gel electrophoresis. So this is a question about how heterogeneous can a sample be. Then I have a question here from Michael from Germany, who is asking a question about the size of nanoparticles that can be analyzed. So he's looking at a size range 50 to 100 nanometers. Then I have a question here from Ankit from India about limit of absorbance or concentration to use in AUC.
Okay,
maybe you can group this together. Yeah?
Okay, I will go through the questions. Thank you very much, Lutz, for grouping it. Concerning the purity in this paper, I think I know what you mean, corresponds to interaction analysis. If you want to have to analyze a system, A plus B goes to AB, for example, the sample has to be really pure, because otherwise the isotherm you derive is simply not correct. So for interaction analysis, it should be as pure as possible, and 95% is a good starting point. Your question, you want to look for aggregates and recombinant proteins, and when your protein purity is usually 90 to 97%, this is no problem, because you get a size distribution showing you that you, for example, have only, let's say, 91% monomer in there, or 91% of recombinant protein, and the rest is host cell protein, for example, then you get this result. But you may not use this sample to look for interaction with a target, for example. But for size distribution, there's no limit for the purity of the sample. Okay. Going to the nanoparticle question. You can analyze nanoparticles in that size range. You simply have to lower the size, the speed of your centrifuge, because usually nanoparticles are much more dense than the than proteins. So you have a V bar of let's say 0.2 only, or even lower. So it depends, of course, on the material. But if you have the nanoparticles, let's say, consisting of a metal core and a shell of a polymer, then the size range is usually okay. You may start with 3000 RPM. You should not go lower, because then your meniscus is not sharp anymore. But you can also densify your solution. But when starting at 3000 RPM, I think it should be feasible. I have also analyzed, it was a colloid of concrete, so to say, and the particle size was even a little bit higher. What you should be aware of is if you use nanoparticles, they may impact on the bottom of your cell, so you should not use the EPON centerpieces, but go to aluminium or titanium centerpieces, because the nanoparticles, if they consist of metal, they are slammed into the bottom of your cell, and you may never be able to recover them. Okay, then I will go to the question with the concentration limit. For the absorbance, the lower limit, in my experience, is it should be not lower than 0.05 mg per mil, so 50 microgram per milliliter. You may switch to 230 nanometers and still be able to record a good absorbance pattern. The upper limit, I would set a maximum at 0.9 or 0.95 OD, because at 1, the photomedia is not the newest one. You already have a quite high signal to noise ratio. So I would recommend not to go much above 0.9% and OD at 280 nanometers, corresponding roughly to, for most monoclonals, to 0.5 or 0.6 mg per mil.
I add two more questions here about the concentration. So one question was here coming from Arthur from the UK, who wants to study monoclonal antibody up to 100 mg per ml, and he's asking a question how you can do it by working off peak of the absorption. Yeah, I tried it using a protein concentration of, I think it was 120 mg per mil, and used 300 nanometers, so really off the peak. But in my experience, it's really difficult because you work in the peak shoulder of your absorbance, and you may hit sometimes 301, 299 nanometers, even if you do not change the wavelength. So for my work, I introduced, so to say, a concentration limit of 80 mg per mil, and also tell all my colleagues that there's a limit at 80 mg per mil, and I will not go beyond this. You may change the centerpiece, of course, and use a 1 millimeter centerpiece, but they are really tricky to put into the cell. I have to admit I damaged one. Yeah. Okay, so I asked the next question here. This is, I think it's interesting for all the biopharma participants here. I got a question from Switzerland from Egon, who is asking whether you are using the AUC more for quality control purpose or in the process of drug development. I would like to add maybe the validation of results of other methods might also be a topic here.
Yeah, of course, AUC is hardly to validate. We use it in the very beginning of a project to give a target range, so to say, for the size exclusion chromatography. We do not use it as a regular check, which was sometimes suggested. So to check regularly if the size exclusion still works. You can do this, but usually you have elaborated a quite good size exclusion protocol and only use this to in the beginning to give a start range. And then, of course, we have this comparability exercises during a project where we have to include it, but we do not use it for the regular quality of control, of course. For the drug development in the early stage, the AUC is really useful because you can already give information about candidates, for example, regarding the second virial coefficient. You can also check if your cell lines produce, for example, 100% binding competent material. So this is really, we use it in early drug development and also in the development of the size exclusion method if once the candidate protein has been identified, but not so much for quality of control.
Okay, thank you. I have some more questions here, but those questions are pretty much detailed application questions. So I think that this webinar may be just too short to answer those in detail. So I have the name here of the people asking these application questions, and
yeah, you may hand out my email address for questions afterwards.
We will reach out to you for the application questions and can organize a call here. So I think it's better to do that offline and not going into application detail or at the end of this 1 hour. So I think it's a good time to close the webinar, the call here. I'm available. I continue to be available. If some more questions come up, just contact Beckman Coulter to organize a contact here to get questions answered, additional information, whatever information you need about AUC or centrifugation in general or other techniques or methods. So we're open to have a contact. So when more questions come up, just contact us. I would like to thank Alexander for his presentation, for making himself available and getting approval here for doing a webinar outside a biopharma company, which is not so easy. So very well appreciated, Alexander. And thank you very much for giving the presentation here. And to the audience also, thank you very much for listening. And as I said, if any questions come up, just contact us. Yeah? So I'm closing the call now, and thank you very much for your attention. Okay, goodbye.
Thank you. Bye.